Hello everyone, welcome to another video tutorial. So in this video lecture we will discuss about the chromatography. So now let's start with the introductory part. So the chromatography is defined as an important technique used for the separation and identification of the mixture. So basically it is a laboratory technique used for the separation and identification and also for the purification of different types of components present in a mixture. So based on the different methods, the chromatography is of many types like paper chromatography, column chromatography, thin layer chromatography and many other methods. Now, the chromatography word is made up of two words which are chroma and graphene. So, chroma and graphene both are Greek words. Here the word chroma meaning color and the word graphene indicates to write. So, this combination of words is used for the invention of the technique which is used for the separation of pigments. So initially this method was used for separating pigments from a particular mixture of different pigments, but nowadays it is also used for the purification of different components and identification of components from a mixture. Now let's understand about the history and discovery about the method of chromatography. So basically the method of chromatography was first discovered in Russia by the Italian-born scientist named as Michael Sweet in 1900. So he developed the technique for the separation of plant pigments. We know that plants are also a living organisms which have different types of cellular pigments like carotenoids, chlorophyll and many other pigments which help them to perform photosynthesis and for the colorization of flowers and many other functions. So to separate these pigments, Michael Sweet invented a method which is called as chromatography, and he named the technique as chromatography because it is used to separate the pigments of the plants. After that, during 1930s and 1940s, there are different types of chromatography were developed by different researchers and scientists from all over the world, and these methods include paper chromatography, gas chromatography and many other methods. So, chromatography technique was developed as a result of the work of two scientists named as Arthur John Porter Martin and another one named as Richard Lawrence Millington Sienge during the era of 1940s and 1950s, and they also won and awarded by the Nobel Prize in Chemistry for their discovery. So this is the history about the chromatography. So before moving to different types of chromatography methods and procedure of different methods of chromatography, let's understand about the principle of chromatography. So basically, the chromatography is defined as a separation method which is used to separate and to identify different pigments or different components present in the sample mixture. Now in this method, the analyte is combined within the liquid or gaseous mobile phase which is pumped through a stationary phase. So here in this method there are two phases. The first one is mobile phase which can be liquid or gaseous, and the another one is stationary phase. So here the mobile phase have the ability to move and it moves in the stationary phase, and the analyte is defined as the pigment which is needed from the mixture. So this component of the analyte interact differently within these two phases which are mobile phase and stationary phase respectively. So here these, depending on their polarity, these two phases or these components spend more or less time interacting with the stationary phase, and they are thus retarded to greater or lesser extent. Now this leads to the separation of different pigments present in the mixture or present in the sample. And here each sample component elute from the stationary phase at a specific time which is called its retention time. So on the basis of their differentiation, some compounds stay longer in the stationary phases and some other components pass rapidly, which move rapidly from the stationary phase, and that's why they are differentiated into different bands in the method of paper chromatography, and also in the method of other chromatography they are differentiated or separated into different pigments. So this is the principle used for the chromatography methods. So now let's understand about the basis or the terms used in the method of chromatography technique. So basically, as we have discussed in our earlier slide, that there are two different types of phases used in the chromatography method, and these phases are termed as stationary phase and the another one is mobile phase. So here the stationary phase is the substance which is fixed in the place for the chromatography, like silica layer or the chromatography paper in the method of paper chromatography. Now the another phase is the mobile phase, which have the ability to move in the definite direction. So this mobile phase can be liquid in the process of capillary electrochromatography, it can be gas in the case of gas electrochromatography, or it can be fluid also. The third phase is the separated molecules, which are separated during the method of chromatography technique. Now there are some other terms which are related to chromatography, like analyte, which is the substance to be separated in the chromatography method, or we can define the analyte as the substance which is needed from the mixture. Now the chromatograph or aerograph is the instrument that enables the sophisticated separation, or the chromatograph is the instrument which is used to separate these pigments. Now there are two other terms named as eluent and eluate. So here the eluent is sometimes also spelled as eluent, which is the solvent or the mixture of solvent used for the elution chromatography, and the eluate is the mixture of solute or solvent existing in the column in the process of column chromatography. So beside these, there are several other terms related to the chromatography technique, like chromatogram, bonded phases, electrophic series, and solute, solvent, detector, which is the instrument used for the quantitative and qualitative detection of the analytes after the separation of these pigments. So these are the basis of the chromatography technique, which are named as stationary phase, mobile phase, and separated molecules. Now the chromatography is differentiated into different types on the basis of methods adopted for the chromatography. These types include adsorption chromatography, thin layer chromatography, column chromatography, and partition chromatography. So let's start with the first type of chromatography, which is adsorption chromatography. So the first type is the adsorption chromatography, and it is the process where different compounds are adsorbed on the absorbent to different degrees according to their absorptivity. So as we have discussed that there can be different types of components or pigments present in the mixture or the sample. Now the degree of absorbability or different types of components can be different, and on the basis of different degrees of their adsorbability they are separated on the different pigments. So this method is the common method used in industries for the chromatography. Now the another method include thin layer chromatography. So here it is also called as TLC, and in this method the mixture of substances or components is separated into its pigments with the help of a glass plate, which is coated with a very thin layer of adsorbent, and that's why it is named as thin layer chromatography. And the absorbent includes like silica gel and alumina, which are used in this method. So here the plate is named as chrome plate, which is used in this method, and the solution of mixture is to be separated is applied in a small spot as the distance of 2 cm above one end of the plate. And these different components of the mixture are moved to the different heights, and that's why they are separated. Now the another type of chromatography include column chromatography. So this is the technique which is used to separate the components of the mixture with the use of a column of a suitable adsorbent, and here the adsorbent is packed in a glass tube and the mixture is placed on the top of this column, and a eluent is used to flow down the column slowly. Now here we know that there are different degree of adsorption for different components present in the sample mixture. So according to their degree of differentiation, they are separated into different components, and thus this chromatography method takes place. Now the fourth method include partition chromatography, and the most common example for this chromatography includes paper chromatography. So this method also includes two phases named as stationary phase, and the another one is mobile phase. So in this method a chromatography paper is used as a stationary phase and a mixture of solvents or a mixture of different pigments is used as a mobile phase. So here there is a spot made on the base of the chromatographic paper with the mixture, and the solvent rises up in this paper. So as we have discussed that according to the different degree of these different components, they have different retention on, or they move to the different heights in the chromatographic paper, and that's why the components or the pigments are thus separated at different heights in the chromatographic paper. So these are the different types of the chromatography. Besides this, there are different chromatography methods also used in the industrial field, also in different other fields, named as planar chromatography, thin layer chromatography, ion exchange chromatography, gas chromatography, liquid chromatography, and many others. Now let's discuss about the importance of chromatography. So this method is very important and widely used to separate and isolate different molecules. In the field of molecular biology, the chromatography is used to separate and to isolate different types of proteins and other biomolecules from the sample, and it is also used for the purification of proteins from the sample which is having different types of biomolecules. Now the chromatography is also used in the pharmaceutical industry and food industry to separate different types of food pigments also, and in the pharmaceutical industry it is used to separate different pigments or different components from the mixture of components to find the desired components from the mixture. So these are the importance of chromatographic methods in the field of biomolecular biology and some other fields. So in this video we have discussed about the chromatography, different types of chromatography, and importance of chromatography in biological field. Thank you.